Hey, GS112, welcome to week 9. This will actually be the last week of video lectures, since that very last week, which actually corresponds to next week, which is week 10, is going to be kind of a sort of like a finals week. It looks like there is no final time necessarily scheduled for the class. So and just to kind of let you know what are going to happen in these final 2 weeks, like I said, this is the last week of video lectures. Oh, by the way, say hi to my friend Steve. He's not looking so good right now. You'll find out what's wrong with Steve in just a little bit. You'll never find out what's wrong with me. So I wanted to mention that in week 9, a couple of things that you should know about. First of all, that very last, this Friday of week 9 is going to be when your reading assignment is due. Pretty much you just read that last chapter of the T-Rex book. And do a reading assignment. That should be found on this week's modules. Monday of next week, that final week, is going to be, your homework will be due. As normal, I'll post answers to all those homework questions. In preparation for exam 3 on Wednesday, keep in mind exam 3 is going to not be cumulative. It's just going to be any kind of item of new information that we've studied since the last exam. Other things to know about. Oh, also, this week there won't be another lab activity. So my idea is to kind of do it like this, so that final day of registered classes, which I think is next Thursday on June 11th, that hopefully everybody will be able to get a chance to kind of work on all their back work and everything submitted. If there's any question as to what I have for already, accessible for you guys, I know some of you, I'm really sorry. It's been really frustrating with things saying that they're missing when they're not really missing, and it's a huge pain. Don't worry, it's as much of a pain for me as it is for you. Maybe more so? I don't know. So anyway, keep that in mind. I don't think I have anything else to talk to you about. Oh, by the way, this particular week I decided to do what I'm calling the Products of Science week. Keep in mind, this was supposed to be the week where you did all your group presentations, but there's no groups and there's no presentations. So I kind of came up with what I called the good, the bad, and the ugly of science. So these are the actual products that have come out of the process of doing science. The very first one we're going to be talking about today, all about antibiotics and kind of the early history of disease, and how we've come to understand disease and understand germ theory and stuff like that. Me being a chemist, it's something that I always kind of wanted to learn a little bit more about, so I did that. On Wednesday, we're going to hear about nuclear weapons. And that's kind of the quote-unquote bad. We'll actually find out that everything that kind of went into building nuclear weapons wasn't all bad. In fact, there's some really promising things that could actually come out of that research. And then the ugly, which will be on Friday, our last video lecture. I just decided to do something kind of fun. Those of you that know me well enough know that my dad is an insane Sasquatch researcher, so I kind of decided to do that as my pseudoscience. I should mention that a few of you sent some really awesome ideas for various topics that I could have talked about. So what I'm going to do, or if I already might have done for you, is I'm going to send you some kind of peer-reviewed sites for those particular topics. And thank you for submitting them. Unfortunately, we just couldn't fit them in once I realized that there was only this week, basically. So anyway, I hope that you guys have had a good time. Hopefully the fun will continue just a little bit, and I will talk to you soon.
Perhaps you've seen this on YouTube. It's a fire tornado. It's two of nature's most horrifying elements jammed together in one package. To find out how and when these things occur, I asked Marino DiMarzo of the University of Maryland's Department of Fire Protection Engineering to explain them to us, and he did us one better. He agreed to make one for us in his lab.
The fire tornadoes form because in a fire there is a substantial updraft due to the fact that the flame is hot and wants to rise. And if there is any rotational effect in that, that effect amplifies the flame and stretches it. And as it stretches, the flame becomes like a column and becomes much more intense, and at that point the tornado feeds on itself. In nature they're very rare. They are caused because essentially there is a certain point in these random motions, it gets organized in a rotational fashion. And then once it gets to that point, the fire tornado starts. But that's a random event. In fact, they are very uncommon. The fire tornado is much more dangerous than normal fires because the combustion rate is much higher, and as a result of that, the radiation that comes out of the flame is much higher, and whatever is around the fire gets this enhanced amount of heat that can cause trouble.
The real question is, after we've gone through all these things having to do with science that we've been talking about all term, what are some products of that scientific process that we've been introduced to? Well, the one thing we're going to talk about today, which I say the good. I can't think of anyone that doesn't think that these things are good, especially when I explained to you kind of what was going on a mere 100 years ago before their invention. These are chemical compounds that specifically kill only bacterial infections. And this is kind of like, this was the big blockbuster one back in World War II times, which I'm sure you've heard of, but there's just a chemical structure. You guys should know that each one of these letters represents a particular element. So like, for instance, N corresponds to nitrogen, S is sulfur, C is carbon, O is oxygen. And then you'll notice that the way that organic chemists oftentimes will abbreviate things is they'll actually leave out carbons and hydrogens. So anywhere you see kind of like a corner here, that actually is a carbon. All these like dashes and solid wedges and things like that, they have something to do with kind of how these things are held in three-dimensional space. So you should definitely take a chemistry class from me, and we'll learn about all the fun of organic chemistry. And also you should probably know that, like, for instance here, carbon always bonds 4 times, so there's actually a hydrogen sticking out at us. But again, don't need to know all that stuff to understand today's talk. So the first question you might have, especially if you're not associated that much with science, is what the heck are bacteria? Well, here is a picture of bacteria, and it turns out they're not these big giant cells here. This thing right here with the little nucleus in the middle, this is actually a cheek cell. And then covering this cheek cell are all these little tiny dots. They almost look like little tubes. So dots are usually called a coccus. You might have heard of staphylococcus. That's what the coccus corresponds to, the fact they're like little tiny spheres. Also, there's like tube-like bacteria that are called bacilli, or bacillus singular. There's also, turns out there's none in this picture, but we'll actually talk about a spiral type, which is called a spirochete type of bacteria. But again, you don't need to know any of that. The fact is that we have all of these little tiny living things that are actually on top of our other cellular living things. These big giant cells are called eukaryotic cells, which are gigantic compared to these little tiny bacteria. So why should we care about bacteria? Well, there's good and bad types, but we're going to be talking specifically about the bad types today. Turns out that these little tiny, almost submicroscopic things just absolutely ravaged the world's population up until about even just 100 years ago. So I just kind of chose a couple of both artists' renditions, which will kind of be on the left, and then also what's called a scanning electron microscope. So these are actual pictures of these little things. And I actually learned something. I learned that they can actually colorize electron microscopy slides now, which is kind of interesting. But anyway, this particular one is Yersinia pestis. You might imagine from the name, it's not something you really want to ingest. This is the cause of the Black Plague. And of course, we talked about, like, for instance, in the Middle Ages, some of the horrible diseases that came through, talking about the plague, and those were kind of like considered the Dark Ages because science wasn't really a part of the picture back then. I did look up a couple of pictures of actual disease, but I didn't want people to vomit on their expensive computers, so I didn't include them in the PowerPoint presentation. If you want to see them, just look up the genus species that I gave you on the last slide and eat your heart out. Blech. Another really interesting looking bacteria is this Treponema pallidum, and this one actually causes syphilis. Which, if you look in the history of anywhere from like 1000 A.D. to about 1800, you'll hear all kinds of different stories of insanity caused by syphilis in famous people. For instance, like Napoleon was thought to have died of syphilis. One funny thing about the history of syphilis, typically this was something that was like, for instance, the Polish called it the German disease, the Germans called it the Polish disease, the Italians called it the French disease, even though it was everywhere. And so it's just kind of funny that everybody else blamed everybody else, even back in the day. So this is actually the bacteria that causes syphilis. And just for your own knowledge, there's all kinds of different other bacteria out there. The gangrene is caused by a Clostridium species, cholera, which is a horrible disease where you have just horrible rounds of diarrhea, and usually you become dehydrated, and you often would die from it. Just good old suppurating, which is a great word, which means oozing pus, or purulent means filled with pus, is actually caused by just a really, really common bacteria that's everywhere. This Staphylococcus aureus and a couple of other examples of that. But the point is that way back in the day, before we had antibiotics, it was commonly the case that any infection pretty much ran its course. And if it didn't result in death, it was probably going to cause a whole bunch of suffering and some really horrible things along the way. So, for instance, you might have heard about, like a simple cut to the finger could often be fatal in the olden days, and that was very true. Not to mention, in wars and such, really the only way to avoid any kind of infection was to actually amputate limbs after injuries. But the problem is that we didn't understand antiseptic techniques, so the tools that were being used would actually cause more infections, and it was just a horrible mess. And so, of course, people recognized that these horrible infections were happening, and so in response, there was a whole bunch of quack cures that really weren't based on science, but were really based on kind of anecdotal evidence, and charlatans that would come by and say, hey, I can cure anything with my fancy pills. And so a lot of times these treatments were as brutal as the sickness. And I have one picture which I'll show you in just a second here. And I should mention that sometimes, just kind of by accident, sometimes these homeopathic cures had kind of a small kernel of truth. Like, for instance, the ancient Egyptians used to put like moldy bread on things. Well, it turns out that that's exactly the use of penicillin that we now have. Right. The problem was that there wasn't necessarily a scientific method associated with it. As I mentioned a couple of slides ago, syphilis really ravaged the world's population for quite some time. Here's a picture of what was considered kind of a treatment ward for syphilis. And this is back, I think that says like 1089, so it's a long time ago. But what this person is doing is they're actually taking elemental mercury, which was you've probably seen it in thermometers and such. Is this really cool substance that it's the color of a metal. It is a metal, but it can actually be vaporized. So this poor person has syphilis, and they're treating them with mercury. My friend Steve over here he is. He's got all kinds of syphilitic sores all over him. He's having a bad day, but luckily he's in the mercury ward where they're definitely going to cure him. Not really. So in a lot of these mercury wards, which by the way were kind of a thing for thousands of years, you would drink, apply, or bathe in like mercury, which is a highly toxic substance, right? It's actually a substance now that we're really trying to get away from. But you often, drink something called a calomel syrup, and it was thought that it would cure syphilis. In terms of just kind of the scientific process, not controlling for variables, it was pretty apparent here. What would happen is that one of the kind of pathways of syphilis as it works through you is that you actually are kind of disease free after like a couple of years of being ravaged by the disease, and it has more to do with the bacteria's life cycle, but it was actually thought it was many times due to this mercury treatment. And so it was almost this thing that kind of tricked people into thinking that mercury is working, but really if they had just been left alone, the same thing would have happened and nobody was there to, kind of figure all this out. So Van Leeuwenhoek was one of the first people, actually was the first person to discover bacteria in 1676. And he did this with a primitive microscope. He's actually a really interesting individual. He didn't have much of a formal education, but he was responsible for kind of starting the field of microbiology. The one thing that was not true, even though that we saw in 1676 the presence of bacteria, really its connection to disease wasn't really understood for a really long time. So one of the things I want you to pay attention to as we kind of go through this talk, I want you to pay attention to the years. Kind of keep in the back of your mind, right, 1676, that was really the beginning of the Age of Enlightenment. And I want you to notice, as I show you these other years, how fast things start to happen once we got science on our side. One thing that we take for granted today is this idea that germs cause disease. That was not at all understood even probably 250 years ago. And so one of the things that science did is it started to kind of work against some of the, the celebrities of their times, right? Galen, for instance, had this idea of miasma theory of disease. That kind of goes back to the idea that disease is caused by bad air, right? I think miasma literally translates to bad air. There was also kind of this really odd individual that makes his appearance through much of like early history of science, this man named Paracelsus, who was really kind of he was kind of a charlatan when it really comes down to it, but he was like a big personality, and he was really one that helped to kind of popularize different kinds of theory of disease, usually to get a pretty good paycheck. But finally, Louis comes by in the 1850s, and he was working with wine, and you probably heard of pasteurization, but he was really the first one to kind of recognize that, wait a second, all these like strange microscopic organisms that we've known about for a while, including these little particulates that I don't know if they're even alive, they actually seem to somehow be associated with disease. So this was kind of the beginning of the idea that germs exist, right? And keep in mind, there's all kinds of different types of germs. There's protists, which are kind of like big cells that can cause disease. There's fungal infections. Today, we're only going to be talking about bacterial infections. And of course, we all know about viral infections now. And viruses are actually even smaller than bacteria, much smaller. So this was something that would definitely help to develop germ theory. Here's my man Lewis. Dude, if you got glasses like that, you know that you're going to be a stud. This is Louis Pasteur in his lab. At about the same time that Louis Pasteur was doing his work in France, there were other chemists and other scientists figuring out other things. In this particular case, I call this bacterial carpet bombing, because chemists eventually discovered that certain compounds can be used as quote unquote antiseptics, right? That idea that septic kind of corresponds to infection, and so this is just saying that it is anti-infection, really. So the individual was named Joseph Lister, as a German medical doctor. In 1867, he first uses this substance known as phenol. Keep in mind, we've got carbons on each one of these positions here. And so this was great, right? We had now a way to keep materials or like surgical instruments and stuff like that clean from these little substances that cause germs. But the problem is that once you actually had an infection, you couldn't really do anything. It wasn't like you could just drink phenol. Actually, that was a personal story of mine. I always thought I was much smarter than I actually was. So I remember I was taking a microbiology class, and I had a pair of shoes that always stunk. And I thought, hey, I know what I'll do. I'm going to get rid of all the bacteria that I've been learning about. And I poured phenol into each one of my shoes. I made sure that it was really nicely saturated, and then I put on my shoes and I wore them for about 2 hours afterwards. And I got about second-degree chemical burns all over my feet because one of the reasons that phenol is not used very much anymore is that it's also very toxic to living cells, such as the cells that made my feet. So anyway, I'll never forget good old phenol here. This is just a picture of Joseph Lister, just because, I mean, those sideburns are pretty sweet. Keep in mind that I asked you to look at the time periods we're talking about here, right? So we're talking about diseases that literally ravaged mankind and womankind for, oh, I don't know, thousands of years. As long as we've been around, these diseases have been just absolutely killing us, maiming us, causing suffering without a doubt. And then, all of a sudden, around like the late 1600s, this individual shows up and says, hey, I got these weird specks in my microscope, right? 1850, we've got Pasteur doing his work with germ theory. And suddenly, we, about 10 to 20 years later, have this idea of antiseptics, right? Something to actually be able to kill the bugs that Louis Pasteur found. And like all of this time, right, things are just absolutely getting faster and faster and faster. And so after thousands of years of nothing, all of a sudden we're working our way towards what turns out to be the first antibiotic, which is kind of a mouthful, called arsphenamine. It's a real treatment for syphilis. And by a treatment, I mean it specifically treats the syphilis by attacking the bug or the bacteria that is the cause of it. This is really the first instance of a true antibiotic. It was first isolated by Paul Ehrlich and Hichiro Hada in the early 1900s. And this is actually something I learned. I had no idea that we had an antibiotic this early. I was always under the impression that it was penicillin in, like, for instance, World War II. But no, it turns out that there was actually quite a few antibiotics before that. Just because I'm a chemist, I wanted to include a little bit of structure. Notice this is arsenic, which is also something that's very dangerous to life. And it actually is interesting because even though we've been using this drug for a very, very long time, we actually still don't understand exactly what the biological action is in the body. That's something that's often true of certainly living chemistry or human biology and chemistry. We often don't understand exactly what these things are doing. We just know they work because of the scientific method. And so here we go, the development of penicillin. It truly is a wondrous drug. It completely changed the world. So this was finally a treatment for everyday infections, right? That other one that we just talked about specifically treated syphilis infections, or the Treponema infections, but Treponema is a really unusual bacteria, and it didn't really work for anything else, that particular antibiotic, which is called Salvarsan. So penicillin, right, this had all kinds of possibility for treatment. It seemed to work against quite a few infections. It was discovered by Alexander Fleming in 1928, was literally by accident when he left the lid off of a petri dish, and some moldy bread that he had probably eaten a couple of days before got in. And I say by accident, but the discovery itself was pretty profound in that he noticed, like, hey, wait a second, those, I'm trying to grow this staphylococcus and it's not growing wherever my moldy bread got in. So this, of course, led to mass production of penicillin in World War II. This was truly an engineering miracle, like the ability to just all of a sudden double down and create huge vats of penicillin to work for, or to be able to treat troops in World War II was truly an engineering miracle. I think in 1945, Fleming and two other individuals, I can't remember, won the Nobel Prize in medicine. So the one good thing is that we kind of understand quite a bit of how penicillin works. It turns out that there are cell walls in bacteria that we do not have as human individuals, but bacteria do. And so there's this little protein that has to go from one place to another. And it turns out what penicillin does is it takes this little thing, which is called an acyl group, and kind of sticks itself onto a really important part of that enzyme, and it won't work. So because of that, the bacteria can't build a cell wall, and it explodes, essentially. So of course you're probably aware of the fact that there are just probably tens of thousands of antibiotics now that have all kinds of different structures. I'll show a couple of structures in just a second, but if you're interested in this, you may want to check out this link, and it actually lists out a ton of information about antibiotics, including various classes, what they work against, how they do it biologically, which I found pretty interesting, but I won't drone on about it here, but definitely check it out if you're interested. So we talked about penicillin, right? And you can notice that all the cephalosporin type antibiotics look very similar. That's kind of one thing that's true of chemists. We kind of wait around for various living systems to kind of do most of the work for us, and then we take something and then we alter it in some way, right? So we might take this thing, which is a cephalosporin, also from a fungus. This arrow is just pointing to something called a beta-lactam, which is common to this class of antibiotic. But a chemist's job was often to just kind of take these little groups, like this is called a carboxylic acid functional group, and just kind of stick other ends on there. And it turns out that we can develop entirely new antibiotics quite often by doing that kind of thing. So here's just a couple of different chemical structures for things that you might have taken in your life before. Tetracycline antibiotics, that cyc corresponds to circles. So you notice that tetra corresponds to 4, and then the cycles, and so this guy actually has 4 cycles all kind of fused together. A lot of you probably know people that are allergic to, say, sulfa drugs. So here's an example of what's called a sulfonamide antibiotic. That R group just means it could be just about anything. The important part is right here. Erythromycin, I think my kids were just on there, or one of my kids was just on that not too long ago. They all kind of have this big giant, what's called macrolide structure with a couple of carbohydrates stuck on. But anyway, that's just to kind of give you an idea of all the, just the absolute explosion that was only possible through chemistry, and, kind of the junction of chemistry and medicine and biochemistry is pretty amazing. So one important thing I should leave you with, you probably heard about these resistant bacteria. And so we got to know the job ain't over, right? So the bugs, you're constantly working against this idea of evolution, right? So like you might have heard about methicillin resistant Staphylococcus aureus called MRSA, right? It turns out that there are certain bacteria that as we take more and more antibiotics, they kind of will start to, through an evolutionary pathway, come up with ways around antibiotics. And this is really scary for a lot of people, because you could conceivably, if you're not good about antibiotic use as a citizen, you could actually end up helping to evolve something that is beyond our ability to treat. So new classes of antibiotic are needed. I don't think there's been a new one since like the 1970s. So this is a kind of a war that keeps continuing. Biology is a pretty powerful thing. So there's just the various websites that I looked at. I will talk to you on Wednesday.